everyone. Today we'll be continuing from where we stopped last time. We established that the essence of glycolysis was the production of pyruvate. But the big question comes, what is this pyruvate used for? What is the function of pyruvate, or what's the fate of pyruvate? That is, after forming pyruvate, whether directly from glucose, which is in glycolytic pathway, or from any other source, because there are other sources of pyruvate, which we'll mention in our series later. But whatsoever the source, what is the fate of pyruvate? What can pyruvate be used for or metabolized into? Now, pyruvate has 6 different fates, which we'll be talking about today. It has 6 fates. Now, number 1, following glycolytic pathway, when pyruvate is formed, if it is in aerobic conditions, listen very carefully, if it's in aerobic conditions, the pyruvate is converted to acetyl-CoA. So now in aerobic condition, I want to produce more ATP. So the pyruvate is carried into the mitochondria. And mind you, pyruvate is one of the 3 molecules that can actually pass the mitochondrial membrane. So pyruvate alongside, that is 3 carbon, malate, 4 carbon, and then citrate. Now, so once it is carried into the mitochondria, within the mitochondria, it is converted to acetyl-CoA. That is in the presence of oxygen. Now, in the absence of oxygen, pyruvate is converted to lactate. That's why someone that is having exercise for a long time, prolonged exercise, where it has converted to anaerobic respiration, over a long time, there should be accumulation of lactate. Why? As I keep using up glucose to form pyruvate, those pyruvates are converted to lactate. Now, the third fate of pyruvate is that pyruvate can be converted to oxaloacetate. Now, though that will be in 2 phases, I can use different steps. Now, the first one is that directly I can convert pyruvate to oxaloacetate. Pyruvate is a 3 carbon person, I convert into 4 carbon. So that will be number 3. And then number 4, I can still convert pyruvate to oxaloacetate, but this time around, I will form malic acid first before I form oxaloacetate. So that one will be like 2 steps. We'll look at it in details. Now, another thing I can do is that pyruvate can be converted back to glucose. That's a process called gluconeogenesis. And on other conditions, pyruvate can be converted to amino acid. So let's pick them one after the other so we don't miss it. So we start with the first one. So the first phase of pyruvate is in anaerobic conditions, absence of oxygen. So pyruvate, so that will be conversion of pyruvates to lactate. That is in this case lactic acid. I'm using lactate here because hydrogen ion is usually lost, and this is in the human body, so most times it's usually lost. Pyruvate changes to lactate. And this is actually a reduction reaction. The essence of this reaction is to replenish the used up NAD+. Now remember, in step 6 of glycolytic pathway, NAD was converted to NADH plus H+. So I kept converting NAD to NADH, NAD plus NADH. Now over a while, that if there is no oxygen, the NADH in that step is not carried into electron transport chain to produce more energy. So what does that keep the body at? That means gradually NADH will be increasing, but NAD will be decreasing. Now, you know that step required NAD plus, not NADH. So the way by which the body ensures that the NAD plus that was used to form NADH in step 6 of glycolytic pathway is converted back to form back its original is this guy. And this happens in anaerobic glycolysis. I believe you are following me. So in normal aerobic glycolysis, quite simple, NADH produced is carried into electron transport chain. That is in the presence of oxygen. But when there is no oxygen, absence of oxygen, this NADH has to be converted back to NAD+. And this is the step, pyruvate to lactate. And the name of this enzyme is called lactate dehydrogenase. Now from the name, this is a reversible step. So from the name, you see clearly that it's better named this way. Why? Lactate dehydrogenase, they are removing hydrogen from lactate to give to them. But since it is reversible, the reverse is what is happening here. So that's the first phase, and it happens in the absence of oxygen. Now the second phase of pyruvate is that pyruvate can be converted Please, this happens in the cytosol, in the cytoplasm. Pyruvate can be converted to acetyl-CoA. And like I said before, this happens in the presence of oxygen. So that's aerobic glycolysis. So whenever we're having aerobic glycolysis, presence of oxygen, pyruvate formed is carried into the mitochondria. So the pyruvate I formed will now be carried from the cytosol, where it was formed from glycolytic pathway, into the mitochondria, where it is converted to acetyl-CoA. You could ask me the question, why is it that it was not converted in the cytosol? It is because acetyl-CoA cannot cross the membrane. Remember I said 3 people can cross: pyruvate, malate, and citrate. So pyruvate has to go in, change to acetyl-CoA. And this step is usually a complex step, but it's something that's quite direct. Now let's look at the things to see. Now first, pyruvate is 3 carbon, whereas acetyl, anytime you hear acetyl, that's 2 carbon. It's a 2 carbon guy. Now you see, amongst others, this was pyruvate, this is now a CoA. That means this addition of CoA. And CoA here is coenzyme A. But the way we write it, we usually write it as CoAsh. The idea of the ash there, let me show you. CoA is H. H here is sulfur. So the particular part of the coenzyme A we are targeting is the sulfur containing part of it. So that's the target. So that's why it's written this way. So why would I change from 3 to 2? It's because there has been loss of CO2. Please take note that carbon is lost or gained as CO2. So that means anytime I change from 3 carbon to 2 carbon, there has been loss of carbon, and that is loss of CO2. That's decarboxylation. Now, the enzyme that catalyzes this step is called pyruvate dehydrogenase complex. Now, this is because this step also changes NAD plus to NADH. So this step is also an oxidative stage, whereby, so this is a complex. It actually still contains pyruvate decarboxylase, because if you see carefully, I am adding CoAsh, I am taking out CO2, at the same time, NAD plus is changing to NADH. That's why the name is a complex. Ideally, the main guy here is the dehydrogenase, which is changing this to this, removing hydrogen from here and adding here. But also, there is removal of CO2, and then there's addition of CoAsh. There are other cofactors that are important. We have TPP, thiamine pyrophosphate, we have lipoic acid. So these guys are important too in this particular step. Please take note, it happens during the presence of oxygen, and then once acetyl-CoA is formed, acetyl-CoA is now carried into Krebs cycle for production of more energy. So that concludes our first 2 fates of pyruvate. Now we go to the third fate of pyruvate. The third fate of pyruvate. Now the third fate of pyruvate is that pyruvate can be converted to oxaloacetate. Of course, some books will mention oxaloacetic acid. It's the same thing. The only difference is that acetate has lost 1 hydrogen, and that's the way we usually see them in animals, because the acid will quickly dissociate to get rid of hydrogen ion. Now, so pyruvate can be converted to oxaloacetate by CO2 fixation. So by just addition of CO2, I can have pyruvate. Remember, pyruvate is how many carbon? 3. Oxaloacetate, as we will learn later, is actually 4 carbon. So this fixation, I told you, addition or removal of carbon is by CO2. So anytime I'm adding carbon to a compound, it's because I'm adding CO2. That's why the carbon changed from 3 to 4. So that's what we have here. And the enzyme, of course, will be called a carboxylase. So I move from pyruvate to oxaloacetate, and the enzyme is called pyruvate carboxylase. And then in this particular step, like we said, CO2 is being added. In this particular step, CO2 is being added. But what else is important about this particular step we are seeing here? What else is important about this step we are seeing here? You realize that from mere glance of this step, I see something is changing from 3 carbon and it's moving to how many carbon? You see it very right. It's moving to 4 carbon. Now, another thing that's worthy of note about this is, why am I forming oxaloacetate? Now the idea is that pyruvate will start forming oxaloacetate after a long while. That's when glycolysis keeps happening, and I keep forming from glycolysis, that is, glucose will be changed to pyruvate. If it's in aerobic conditions, pyruvate will be changed to what? Acetyl-CoA. You got it right. Now look at the problem. Acetyl-CoA will enter Krebs cycle, but usually it goes in there to meet oxaloacetate. So every single time acetyl-CoA binds with oxaloacetate, as I keep forming my pyruvate, so every single time I'm having glucose changed to pyruvate, the pyruvate form is changed to acetyl-CoA, and the acetyl-CoA joins with oxaloacetate to do Krebs cycle. Now look at where the problem is. As I keep forming this guy and forming this guy, with time, the OAA will be decreasing. So a way I replenish the OAA in the body is by, as time goes on, the pyruvate will not just form acetyl-CoA, the pyruvate will change directly to form OAA. And this kind of reactions are usually called anaplerotic reactions. It's one of the things we'll talk about when we are done talking about the Krebs cycle. So anaplerotic reactions. Okay, so right here, anytime you have to do with carbon fixation, please, biotin is an important cofactor. And aside from biotin, another person that is an important cofactor is acetyl-CoA. Why would you think acetyl-CoA is important? As acetyl-CoA keeps increasing too much, the body will be like, instead of me forming only acetyl-CoA and this guy is going down, at least as I'm forming this person, let me also be forming who he will join with. So that's why pyruvate will change to oxaloacetate. And like I said, it's called anaplerotic reactions. So now let's go to the food phase. This is yet another anaplerotic reaction whereby pyruvate is converted to oxaloacetate, but this time around it is using malic formation. So that is malic here, malic acid, malate. Malic acid, that is malate. So first I want to form OAA, but first I convert from pyruvate first to malate, and from malate I now form OAA. And it's quite simple. Malate is still a 4-carbon guy, so that means when I change from pyruvate to malic acid, I'm still adding a CO2. So I'm changing, like I said here, 3-carbon to 4-carbon. That's why I'm adding CO2. Something similar happens here, 3-carbon to 4-carbon. The only thing. Alright, so like I said, here we're changing from 3-carbon to 4-carbon. So addition of CO2, and then also NADPH is converted to NAD+. So it's actually a reduction reaction. So I'm reducing to form malic acid. But the malic acid does form is now changed to form OAA. And that's usually an enzyme called malate dehydrogenase. And right here, NAD+ is changed to NADH+H+. Here you can see carefully, please, these are two different persons. NADPH is different from NADH, please. Okay, so please, you realize that pyruvate can be converted to OAA, but this time around, I'm changing it to malate, and the malate is what forms the oxaloacetate by malate dehydrogenase. Now, this reaction, just like the third reaction, are both called anaplerotic reactions. They are filling up reactions. They fill up the intermediates of the Krebs cycle. So they are one of the two key reactions in anaplerotic reactions, or filling up reactions of the Krebs cycle. So while here it was a straight pathway, pyruvate carboxylase adding CO2 in the presence of biotin and acetyl-CoA, right here pyruvate is changed first to malate, and malate is converted to oxaloacetate. Now the fifth fate of pyruvate is that pyruvate can be converted to glucose, and this is a process called gluconeogenesis. So now most times, of course, I had earlier mentioned that pyruvate is not only formed from glycolysis, so I can form it from other sources, especially in period of starvation, when the person is out of food and you're not eating. At those moments, you get glucose from pyruvate from other sources, and then at first, and when you've used up your glycogen storage, what the body has to do in those situations is that it will convert amino acids, so it will break down proteins, convert the amino acids to give you glucose. But how does it achieve this? Most times, its entry point is that all those things are converted either to pyruvate or acetyl-CoA. But in any condition that are converted to pyruvate, the pyruvate will be converted to glucose, and it's called gluconeogenesis. Alright, so we'll go into step 6. In step 6, which is the last step, pyruvate can be transaminated to give amino acids. So I can use him to give amino acids. Now, what's the idea here? Now, after we're done taking carbohydrate food, you understand the taking in of food. Back when we first learned, we were taught that proteins are the bodybuilding food. But then when I take in carbohydrates, you still see that to some extent you have some level of bodybuilding. That's because as you keep using your carbohydrates, your glucose for energy, a part of it will be shunted. Part of the pyruvate will be shunted to form amino acids that can be used to build up your body. So that is that about the fate of pyruvate. So you see carefully, in summary, that pyruvate has different fates. Pyruvate form from glycolysis majorly or other pathways. Can either, number one, if it was in the presence of oxygen, it will change to acetyl-CoA. In the absence of oxygen, it will form lactates. Now, on other conditions, in the presence of oxygen, as I keep forming acetyl-CoA and it has accumulated a lot, the next thing that you realize the more I do Krebs cycle, the more oxalic acetate diminishes. And because of that, we now use the pyruvate to also form oxalic acetate through step 3, through 3 and 4. That's either direct carbon CO2 fixation, or also it could also be indirect, whereby I form malic acid first before I form OA. And then pyruvate can be converted to glucose, gluconeogenesis. It can also be transaminated to give us amino acids. Thank you very much for joining us.